Hello friends, in my previous videos we discussed about an overview of light and dark reactions in photosynthesis. This video is exclusively regarding the light dependent reactions or more commonly known as the light reactions. We start with this diagram, and if you remember I told you that these are two photosystems, and where they are located inside the chloroplast and in the membrane of a thylakoid. So they are located in the thylakoid membrane inside the chloroplast. So here these light reactions are going on. What happens first of all? Absorption of light by chlorophyll and other pigments will occur. Here light is coming and absorption of light by chlorophyll and other pigments is occurring. On absorbing a photon of light, what happens? One electron of chlorophyll goes to a higher orbital and gains more potential energy. This is known as the excited state. This chlorophyll goes into its excited state. But this excited state is unstable and hence it goes back to its original orbital in 1/1000th of a second. Okay, so this molecule is excited. It will go to a higher orbital and in a billionth of a second it will come back. While doing so, energy is transferred from one pigment molecule to other. See, one pigment molecule to other and so on. And lastly, it reaches a pair of chlorophyll molecule in the reaction center. Look at the diagram again. This is the reaction center and this is a special pair of chlorophyll A. So here it will reach finally. After this chlorophyll A is excited, because of its special properties, it transfers energy to primary electron acceptor. This is the primary electron acceptor. This is the electron. It goes to primary electron acceptor. After this series of electrons and then chlorophyll A and then primary electron acceptor. Okay. After once it itself gets excited, the primary electron acceptor thus gets reduced, obviously. So, I hope this is clear now. What happens next? Splitting of water occurs. Coming back to the diagram, splitting of water is occurring here. And what happens after that? Resulting in two H ions and one oxygen atom. Okay, this process is catalyzed by an enzyme P680. It will become P680 plus. What is P680? We'll study a little bit later. But in photosystem 2, first of all, photosystem 2 is involved and then photosystem 1. So, the special pair of chlorophyll A here is P680. So, it becomes P680 plus, gets electrons one by one, each electron replacing one, transferred to the primary electron acceptor. The hydrogen ions are released into the thylakoid space, interior of thylakoid. Looking at the diagram again, the hydrogen ions, this is the space obviously. Here they are released, the hydrogen ions. Okay. The oxygen atom immediately combines with an oxygen atom generated by the splitting of another water molecule. So, two oxygen atoms will combine forming O2. O2 here in the diagram. Okay. Photo-excited electrons, they go from photosystem 2 to photosystem 1 via an electron transport chain. See, photosystem 2 to photosystem 1, they are going like this, and this is the electron transport chain. This is the electron transport chain. This contains what? This electron transport chain will contain what? Electron carrier, plastoquinine. We symbolize it as PQ. What is this? It is a cytochrome complex. and plastocyanin symbolized as PC, which is a protein. So electron transport chain, what is this? It is a series of redox reactions. In this process, ATP is generated. ATP is generated over here. Free energy is released and used to pump hydrogen ions into the thylakoid space. Okay. So, what is this? Proton gradient. It is generated. Okay. Now, similarly, chlorophyll A is excited by light photons in photosystem 1. Now, photosystem 1 comes into action. We call it as P700, which then becomes charged P700 plus, and this will accept electron. Okay, looking at the diagram. This is the light energy. This is P700. It is getting excited like this. Now, second electron transport chain starts through ferredoxin. Here, ferredoxin is there, which is a protein. I've given you the diagram again over here. This is photosystem 1 and this is ferredoxin. The electron transport chain is going on and no proton gradient is in this chain and no ATP. We don't see, here we see the ATP, here we see no ATP. Okay. Electron goes from ferredoxin to NADP in the presence of NADP reductase enzyme. 2 electrons are required. From ferredoxin to, in the presence of NADP reductase, forms NADPH. Okay. Electrons in NADPH are at a higher energy level than they are in water, where they started. So, they are more readily available for the reactions of Calvin cycle. Okay. This process also removes an hydrogen ion from the stroma. Remember, hydrogen ion was released here in the stroma. So this reaction will take up an hydrogen ion. Have a look at this diagram again, which we discussed in the previous videos. We discussed that these are the light reactions and these are the dark reactions or light independent reactions. The light reactions and dark reactions both go side by side. The light reactions occur in the thylakoid and the dark reaction in the stroma. In this video, we are exclusively discussing only the light reactions and we are not discussing these dark reactions. So, we end up by saying that they are now readily available for the reactions of the Calvin cycle. See, NADP generated is used, this NADP is used in the Calvin cycle. So, the light reactions, they give NADP, which is used, and ATP also, which is used in Calvin cycle. This electron transport chain, this and this is non-cyclic. Okay. Sometimes a cyclic flow of electrons too. This is cyclic flow. Previously, we discussed photosystem 2 and photosystem 1 both come into action. First, photosystem 2 is involved and then photosystem 1. But when we have a cyclic flow of electrons, photosystem 2 has got no role. And what actually happens is, this cyclic flow is an alternate pathway. Photosystem 1 is used only and not photosystem 2. It is actually short circuiting. What happens from ferredoxin, the electrons go back to cytochrome complex. From ferredoxin, they go back to cytochrome complex, then via plastocyanin to P700 in PS1, no NADPH reduction and no release of oxygen, but ATP is generated. And where this occurs? In many green sulfur bacteria, cyanobacteria, and in plants growing in low light. this is actually an adaptation in C4 plants also, which we will study later. Okay, so this was all about photosystem 1, photosystem 2, cyclic flow of electrons, the light dependent reactions. Now we study about what actually are these photosystems. So we come back to the diagram of this thylakoid membrane. A reaction center complex surrounded by light harvesting complexes. The light harvesting complexes, these are the light harvesting complexes, and this is the reaction center complex. This whole thing is known as photosystem. Now there are two types, photosystem 2 and photosystem 1. Photosystem 2 was discovered later and photosystem 1 was discovered first. Hence they are named so. But when we study those reactions, photosystem 2 comes in action first and photosystem 1 comes in action later. So, this photosystem 2 has a reaction center chlorophyll A and it is known as P680 in PS2 because it works best at a wavelength of 680 nanometers in red part of spectrum, while P700 is in photosystem 1 and it works best in far red light. P680 and 700 are actually two types of chlorophyll A pigments. What are they actually? 680 and 700? They are two types of chlorophyll A pigments and are very identical to each other. Only their associated proteins are different. So this was all about the light independent reactions. Thank you for watching the video.